Hi guys, I'm Mithila Rajapakse. Welcome to our YouTube channel Contraceptive. So this would be the final video from the section Reproduction, which comes under unit number 5, Animal Form and Function. So let's start off the lesson. Taking a look into the contents of this video. First we have the maternal immune tolerances of the embryo and fetus. And next we'll be starting about the process of parturition, then the process of lactation, and under that the composition of human milk and significance of breastfeeding, and then we'll be studying about the birth control methods, under that common temporary birth control methods and the surgical sterilization methods. They are the preventive methods of gamete release, and next the process of abortion. And next we'll be studying about the sexually transmitted diseases or the STDs. Next the detecting disorders during pregnancy. And then we'll be studying about infertility, and under that we also will be studying about the modern reproductive technology for resolving infertility problems. And we'll be learning some additional facts for extra reference. And we'll be studying about abortion, the proper use of a condom, and proper use of oral contraceptives such as Postino 2. So let's start off the lesson. Maternal immune tolerances of the embryo and fetus. During pregnancy, the overall regulation of the mother's immune system changes. These changes allow the mother to keep the embryo in her uterus without rejecting as a foreign body, even though half the embryo's genes are inherited from the father, and many chemical markers on the surface of the embryo are foreign to the mother. Now, naturally in our body we have defense mechanisms. So the body detects any foreign bodies once it enters the body of the human. So now here the new embryo has a set of paternal genes. Now these are being inherited from the father, and they are new to the mother's body's immune system. So here the immune tolerance is developed, so that there will be no rejection of the embryo by the mother's body. So that is the maternal immune tolerance of the embryo and the fetus. So it's the same with the fetus. The fetus, in the genome of the fetus is different from the mother's genome. So the same happens in the other way as well, so that there will be no rejection occurring between the fetus and the mother. Now next we'll be studying about the process of parturition. First, the childbirth begins with the labor. The labor is a series of strong and rhythmic uterine contractions that push the fetus and placenta out of the body. Now that is the process of parturition. Uterine contractions, in the sense these are the smooth muscles of the uterus. They contract and they push the fetus and placenta out of the body. When labor begins, local regulators, or the prostaglandins and hormones, mainly estradiol and oxytocin, induce and regulate further contractions of the uterus. Now these prostaglandins and the estradiol and oxytocin induce and regulate the contractions of the uterine wall. This is a positive feedback mechanism, as uterine contractions stimulate secretion of oxytocin, which stimulates further contractions of the uterus. So with the increase in contractions of the uterine wall, the secretion of oxytocin increase. Now when the oxytocin increase, the uterine contractions increase. So this occurs continuously in a positive feedback loop. The labor can be divided into three main stages. The first is the thinning and opening up, or the dilation of the cervix. So here, this is the cervix. This region needs to open up so that the baby can pass out. And the second stage is the delivery of the baby. In this stage, continuous and strong contractions force the fetus out of the uterus and expel through the vagina. So that's the process where the fetus comes out of the vagina. And the last step is the delivery of the placenta from the mother's body. So here you can see the placenta is being delivered. So here basically you can see the main steps of parturition. First, the 9-month-old fetus is here, and next the amniotic sac ruptures, and then the cervix widens, and then the fetus is gradually pushed out due to the uterine contractions of the uterine wall, and as a final step the placenta is delivered through the cervix and vagina. Now next, the process of the lactation. The lactation, which is unique to mammals, begins as postnatal care. Lactation is the secretion and ejection of mother's milk from the mammary glands. That's the process of lactation, which is the secretion and ejection of the milk from mammary glands. Lactation is subjected to nervous and hormonal regulation. Now, the lactation process is handled by the nervous system as well as the hormonal system. The main hormone in promoting milk synthesis and secretion is prolactin. Prolactin involves in the synthesis and secretion process. In response to suckling by the newborn baby, which initiates nerve impulses from the touch receptors in the nipples, and decrease in estradiol and progesterone levels in the mother's blood after birth, the hypothalamus sends impulse to the anterior pituitary to secrete prolactin hormone, which stimulates the mammary glands to produce milk. So here, in response to the suckling and decreasing estradiol and progesterone levels in the mother's blood, the hypothalamus sends impulses. So the hypothalamus detects these suckling responses and the decrease in estradiol and progesterone levels. Then the hypothalamus sends impulses to the anterior pituitary, and the anterior in return secretes prolactin hormone. Now, prolactin stimulates the mammary glands to produce the milk. Suckling also stimulates the secretion of oxytocin hormone from the posterior pituitary gland and triggers the release or the ejection of milk from the body. Now, the production of milk is assisted and controlled by prolactin, whereas the release or the ejection of milk from the mammary glands is done by oxytocin, which is produced from the posterior pituitary. This is a positive feedback mechanism, as milk availability encourages continuous suckling. So touch sensation on the nipple and oxytocin release continue further ejecting milk from the mammary gland. So this is also a positive feedback loop. As the touch sensation on the nipple increases, the oxytocin release increases. So with oxytocin, the contraction of the mammary glands increases, and this will result in the further ejection of milk. So here you can see this process in a diagram. First, the stimulation of the nipple by the baby suckling initiates action potentials in sensory neurons that connect with the hypothalamus. So when the suckling is done here, the neurons in connection with the hypothalamus activate and sends the message to the hypothalamus. In response, the hypothalamus stimulates the posterior pituitary to release oxytocin and the anterior pituitary to release prolactin. Now here, the posterior pituitary releases oxytocin and the anterior pituitary releases prolactin. Now prolactin is involved in production of milk, and oxytocin is involved in the ejection or the release of milk from the mammary glands. Oxytocin stimulates milk release from the breast, prolactin stimulates the additional milk production, as I said just now. Next, the composition of human milk and the significance of breastfeeding. During the first few days after birth, the mammary glands secrete a fluid called colostrum until appearance of true milk. Human milk is a sterilized solution that contains nutrients such as lactose, fatty acids, amino acids, minerals, vitamins, and water that are ideal for baby's digestion, brain development, and growth. Now, during the initiation of this milk secretion, initially the colostrum, which is yellowish like color fluid, is secreted, and after that only gradually human milk starts to be released. Now this human milk contains these mentioned components: lactose, fatty acids, amino acids, minerals, vitamins, and water that are ideal for baby's digestion. So these components are in the most simplest forms, like amino acids and fatty acids and lactose. They are simple sugars, so they can be and simple molecules, so that they can be easily digested by the newborn infant. Human milk includes proteins such as casein, lactalbumin, and immunoglobulins. Now they are proteins. Colostrums and true milk provide nutrients for the baby and contain important antibodies that protect the infant. Now, mainly this colostrum contains many antibodies. So this is important in maintaining the defense mechanisms and initiating these defense mechanisms in the body of the newborn baby. So also true milk also provides nutrients and antibodies to the infant. Several types of white blood cells are also present in breast milk, which help the baby to resist microbial infections. Now, still the baby's defense mechanisms are not fully developed. So, these several types of white blood cells which are present in breast milk assist in carrying out defense in the body of the baby. Compared to true milk, colostrum contains less nutrients, less lactose and no fat, but they are adequate for early nutritional needs. Now, they are not as nutritious as the human milk, but they also contain a lesser number of nutrients. So here you can see this first initial 3 days of breastfeeding. This is the colostrum. This yellow color liquid is the colostrum, and here gradually you can see after 25 days this is the human breast milk. So you can see the variation in color. By 25 days it has almost turned to an off-white color from this dark yellow color. So this is the colostrum. Breastfeeding supports optimal infant growth, enhances intellectual development, and fosters mother-infant relations by establishing early and prolonged contact with them. Now, these are some important sense of breastfeeding. Firstly, it promotes optimal infant growth, and then intellectual development. Now, intellectual development of the baby starts with the feeding of the breast milk, and then fosters mother-infant relations. Now, this helps in building up a close and early relationship between the mother and the infant due to the prolonged contact between them. Compared to cow's milk, the fat, iron and the proteins in the breast milk are more readily and easily metabolized. Now, in this powder formed milk, it contains many complex nutrients and molecules that are not easily metabolized. So now in this breast milk, the fat and the ions, proteins, etc. are in simpler forms that can easily be metabolized by the digestive system of the newborn baby, because the digestive system is not totally developed, so it is still under developmental stages. So here the mother's milk contains these nutrients which can be easily metabolized. Lower sodium content of breast milk is more suited to the baby's needs. So this compared to cow's milk, the breast milk contains lower sodium content. The baby is less likely to have allergic reactions to the mother's milk than the milk from another source. So they are less prone to give allergic reactions within the body of the baby. They're talking about the birth control methods. The deliberate prevention of pregnancy is called contraception. This can be achieved in several ways. Now, contraception can be achieved in several ways. Some contraceptive methods prevent gamete development or the release of the gametes. Now, this is one way of achieving contraception, that is preventing gamete development or the releasing of gametes from the gonads. Next is, some contraceptive methods prevent fertilization of the gametes. The gametes are produced, but the fertilization of the male and female gametes is prevented. Some methods are available to prevent implantation of an embryo. Now, after the fertilization also occurs, these methods prevent the implantation of the produced embryo. Unwanted pregnancies are avoided by birth control methods. So that's the contraception where pregnancies are prevented. Common temporary birth control methods. So these are temporary birth control methods. First is the oral contraceptives for females. Most oral contraceptives contain high concentration of synthetic estrogen and progesterone. So these oral contraceptives contain artificially made estrogen and progesterone hormones. High levels of these hormones inhibit GnRH, which is gonadotrophin releasing hormone, released from the hypothalamus. And so FSH and LH secretions from anterior pituitary through negative feedback occurs. Prevention of LH release blocks ovulation. LH blocks the ovulation process. Inhibition of FSH secretion prevents follicle maturation. So FSH is needed for the maturation of follicles. So with the inhibition of FSH, the follicle maturation is prevented. Some oral contraceptives contain only high levels of synthetic progesterone, or you call it progestin, which thickens the cervical mucosa so that it blocks entering sperms to the uterus. When the cervical mucus increases, the path of the sperms will be blocked. So the path of sperms will be blocked, so the sperms won't be able to reach the egg cell to fertilize it. If fertilization occurs, it can interfere with implantation as well. So basically this contains thick cervical mucus. So this implantation process will be hindered due to this layer of mucus. The next option is the use of condoms for males. Now this is a barrier device which prevents the entry of the sperm. This prevents the entry of sperm into the vagina, and the sperms and the semen gets collected in this condom before entering the female reproductive system. The third method is the IUD, or the loop for females. IUD stands for intrauterine device. This is a device placed in the uterus which interferes with the fertilization and prevents implantation of a fertilized ovum. Now this is a small copper instrument, and this interferes with the fertilization and prevents implantation of the fertilized ovum. The fourth method is the Depo-Provera injection for females. The periodic injection of a synthetic progesterone which thickens the cervical mucus and prevents sperm entry. So these are the two functions: thicken cervical mucus and prevent sperm entry. If fertilization occurs, it also prevents implantation by making the endometrium thin. Now the endometrium, with the thinning of the endometrium, the implantation cannot occur in a stable manner, so it prevents implantation. Now this Depo-Provera injection is an intramuscular type of injection. It should be injected to the muscles. Now here you can see some oral contraceptive pills. These are some oral contraceptive pills that can be taken to prevent the unwanted pregnancies. These contain synthetic hormones such as estrogen and progesterone, as said before. And here you can see the condoms. This is the boxes, and these are the packs in which the condoms come, and these are the condoms which needs to be placed over the penis during the ejaculation process. This is the IUD, or the intrauterine device. So this is a copper IUD, and this is LNG IUD. So this is the way that the IUD is placed within the uterus. So here you can see the path of the sperms will be blocked. They cannot pass through this T-shaped like structure because the path is blocked, and in addition implantation also cannot be taken place because the fertilized ovum or zygote cannot move downwards to get implanted. It's being blocked by this. So this IUD can be used for a certain long periods of time, so such as a few years, but the case is there can be inflammatory responses due to this copper like metals which are included in this device. And this is the Depo-Provera intramuscular injection. This is how it looks like. So this is the bottle. You need to inject this into the deep into muscles. Surgical sterilization, or the preventive methods of gamete release. First you have vasectomy for males. Now here this prevents the release of sperms. The vas deferens are cut and ligated to prevent the moving of the sperms up the male reproductive system. Next you have tubal ligation for females. Here this prevents ovum from entering the uterus. The fallopian tubes are tied together so that the path of the ovum is being blocked. Next is abortion. Abortion is the premature termination of a pregnancy in process. So this here after the fertilization of the egg cell occurs, the partially developed zygote is prematurely terminated. So this is the process of abortion in Sri Lanka. Abortion is illegal. So that's the process of abortion. Miscarriage. This is the spontaneous abortion which occurs naturally. That's a natural process which occurs spontaneously. Now this is here this abortion is also done as a surgery to prevent unwanted pregnancies in other countries. Induced abortion is intentionally performed, or surgical or non-surgical processes. Certain drugs can induce abortion non-surgically within the first 7 days after conception. They block progesterone receptors in the uterus, thereby preventing progesterone from maintaining the pregnancy. Now, progesterone is essential for the maintenance of pregnancy. From this, during this induced abortion process, the progesterone receptors in the uterus are prevented. So these receptors won't be functioning properly. So they won't respond to the progesterone levels in the body. So, but progesterone is required to maintain the pregnancy. Now here, so because this progesterone receptors do not detect this progesterone, the pregnancy will be terminated. Now here you can see the process of vasectomy. This is the testis and this is the epididymis, and here you can see the vas deferens are cut. The two vas deferens. Now this is the one vas deferens starting from this one's testis, and this is the other vas deferens starting from the other testis. So both of them are being cut and ligated. So ligation is essential, or else the leaking of this semen, or the sperms produced here will be occurring into the internal environment of the body. So that's why these ends are ligated after being cut. So here the continuous pathway of the sperms are being blocked. So during the ejaculation, the ejaculate does not contain sperms. Only the secretions from the 3 accessory glands of the male reproductive system will be present in the semen. And here you can see the tubal ligation process. Here you can see here and here the fallopian tubes have been cut and ligated. So the ova which are produced from this will not be reaching from these points onwards. So there will be no fertilization, although the sperms enter the female reproductive system. Next we have the sexually transmitted diseases, or the STDs. So here we'll be studying this in a table format. First we'll have the infection, then the pathogen, then the main mode of transmission, and the main symptoms of the infection. First we have gonorrhea. Now gonorrhea is caused by a bacterium known as Neisseria gonorrhoeae, and the main modes of transmission are sexual contact with an infected person, or from mother to child during the birth. The main symptoms of gonorrhea is in males, the burning feeling or the discomfort when passing urine. Now, that's the main feature of gonorrhea. In addition, yellow discharge with pus from the genitourinary tract. Now, this is common for both males and females. In males, burning feeling or discomfort when passing urine occurs. Next, you have syphilis. Now, syphilis is also caused by a bacterium known as Treponema pallidum. This also is transmitted during sexual contact with the infected person, someone who is having syphilis, and also from mother to child during the birth process. The main symptom of syphilis is fever and headache. In addition, you can get rashes, itchy rashes as well. Next you have AIDS, or the acquired immunodeficiency syndrome. This is caused by a virus. This is a viral infection caused by the human immunodeficiency virus. The modes of transmission are during sexual contact with the person having AIDS, or during transfer of body fluids such as blood and serum. So when blood and serum are transfused from one person to another, if the person whose blood and serum is transfused has AIDS, the second person also will be getting AIDS. Use of unsterilized needles. So during use of unsterilized needles, the AIDS virus the HIV virus can be transferred from an infected person to a healthy person. Next, from a mother to fetus or the child during pregnancy, at birth, and during lactation. Now here, during lactation, during breastfeeding also, AIDS can be transferred to the newborn baby in addition to the moment where the birth occurs. So the main symptoms of AIDS are sores or painless ulcers. Now these ulcers are like rashes like areas. They are painless, or any part of the body, vagina, lips, fingers, nipples, you will get these painless ulcers. In addition, you will be having fever, skin rashes, loss of appetite and weight, fever, persistent and dry cough, lymphoma. Lymphoma is a cancer which occurs in the lymphatic system. Now, lymphatic system is involved in providing defense to the body. So, a cancer in the lymphatic system may develop, that is lymphoma. In addition, pneumonia and other diseases resulting from breakdown of the immune system. Now, here you can see the immune system is mainly affected by the AIDS infection. Next, we have genital herpes, or you call it the sexual herpes. The pathogen is the herpes simplex 2 virus. That's a viral infection. The main mode of transmission is through sexual contact, and the main symptoms are itchy, painful sores around genital area, and fever in some cases. So those are the main symptoms for genital herpes. Now here you can see some gonorrhea infected patients symptoms. Here you can see some rashes on the leg, and here you can see yellow discharge or the pus that is released from the penis tip here and here as well. This is a hand of a syphilis infected person, and here also you can see some rashes similar to this but a little lighter. And here you have some symptoms of the AIDS virus. Here you can again see some reddish colored rashes. These are red patches, and in addition you can see these ulcers developing in the mouth lip region. And here you can see some symptoms of genital herpes. Here this is the surface of a penis, and here you can see some rashes again which have been developed due to the sexual herpes. And here these are some rashes in the vaginal area. This is also caused by the genital herpes or the sexual herpes. So here these are these rashes developing. Detecting disorders during pregnancy. Many developmental problems and genetic disorders can be diagnosed during the gestation period. Now, that's the pregnancy period. Ultrasound images can be used to analyze the size and condition of the fetus. So here, through the ultrasound scan images, you can see the development of the baby, and if there are any missing organs or structures of the body, you can be aware of it through the ultrasound images. And also if there are any size abnormalities of the developing organs, those also, all the physical details can be obtained through this ultrasound scan images. Amniocentesis and chorionic villi sampling. Here a needle is used to obtain fetal cells from amniotic fluid or tissue surrounding the embryo. Genetic analysis can be done with this sample. So here amniotic fluid or tissue is obtained from the surrounding of the embryo, because the surrounding of the embryo contains the genetic material of the newborn, of the developing fetus. So through the analysis of this obtained genetic material, you can see if there are any genetic disorders occurring in the resultant in the developing fetus. Newest method is to use a pregnant mother's blood to analyze the genome of the fetus as the mother's blood contains fetal DNA. So here you obtain genetic material from amniotic fluid or the tissue surrounding the embryo. Here you directly obtain the mother's blood. So because the mother's blood contains the genome of the fetus, because you know the mother's body and the fetus's body are connected through the umbilical cord, through the placenta. But all detectable disorders are untreatable when the embryo is in the uterus and cannot be corrected even after birth. So that's the case. Although you identify these diseases, these disorders, they are untreatable when the embryo is in the uterus and cannot be corrected even after birth. However, parents can take informed decisions with the help of these tests. Now that's the advantage of detecting these disorders during pregnancy. When the parents are taking decisions with regards to the fetus or then after the baby is born, knowing these facts will be important in making much more relevant and accurate decisions with regards to the baby. Then talking about infertility. Infertility is the inability to conceive an offspring. Both men and women can have reproductive defects leading to the infertility. Now this is the reason for infertility, that the men and women having reproductive defects. Now here both this man and woman, or the two persons taking part in this reproduction process, should have passed the stage of puberty. So they should be in the fertile stages of their age. The number of couples facing this problem appears to be increasing in the modern society. Certain forms of infertility are treatable. Now here, to be categorized as infertile, the couple should have regular intercourse throughout a period of 1 year, and they also should be in the reproductive age. If all these conditions are satisfied, and if they are unable to conceive an offspring, then you consider to be it as an infertile situation. So here there won't be an offspring being created. Modern reproductive technology for resolving the infertility problem. So now here we have the technology assisting such cases. Some infertility problems are resolved by recent scientific and technological advances. This includes hormone therapy, surgery, and assisted reproductive technology. So here through these fields, this issue of infertility has been addressed through hormone therapy. So by doing some hormonal regulation artificially, this infertility problem can be addressed. In addition, surgeries can be done, and assisted reproductive technology. In hormone therapy, sometimes hormone therapy can increase sperm production in the infertile male or egg production in the infertile female. Now, one cause for infertility can be the reduced production of sperm or egg by the male and the female. So through this hormone therapy, which is done artificially, the sperm production can be increased, or the egg production in the female can be increased. The surgical interventions, the ducts in the reproductive system that are formed improperly or have become blocked can be corrected surgically to resolve this infertility disorder. So sometimes certain structures, or the ducts or the tubes involved in the both male and the female reproductive system may be blocked, or they may have been formed improperly. So they can be corrected by surgical intervention. So then this infertility issue will be addressed. Next you have the assisted reproductive technology. This is the in vitro fertilization process, so you call it the IVF process. In vitro fertilization is a series of procedures used to treat infertility problems and assist. The process involves removal of oocytes from a female ovary and obtaining sperm from a male, and combine the oocyte and the sperm to achieve the fertilization under laboratory conditions. Now here the sperm is obtained from the male and the oocytes are obtained from the female, and the fertilization process is done under laboratory conditions outside the body of the female. The fertilized eggs are then incubated until they reach at least an 8-cell stage, and then these embryos are transferred to the woman's uterus for implantation and to continue its development. So here the fertilization process is assisted artificially under laboratory conditions, and after it reaches about an 8-cell stage, we can assume that the development can take place naturally. So it's then being introduced to the female uterus, the woman's uterus, for implantation, and the developmental process will be continued from there onwards. Conventional IVF needs about 50 and 100,000 of sperms from the male per oocyte in order to achieve the fertilization. So that's the conventional IVF method. This is due to the fact that in IVF, acrosome reaction has to take place and thousands of sperm cells have to be involved. Now here you studied before, acrosome is a specialized lysosome present in the tip of the sperm. Now this is required, these lysosomes, the chemicals in the lysosome are required to penetrate the wall of the egg cell. So here for this penetration process, the contribution of about 50 and 100,000 of sperms are required. So that's why this large number of sperms are introduced per oocyte. So that's the acrosome reaction. Acrosome reaction is the penetration of the wall of the egg cell. Intracytoplasmic sperm injection, or ICSI. This is also a type of in vitro fertilization method which is used to address male infertility. If mature sperm are defective or low in number, a whole sperm or a spermatid nucleus is injected directly into the cytoplasm of an oocyte that has been removed from the woman's ovary. So if the sperms are defective or the sperm count is low, if the semen does not contain the required count, 2 to 5 million per milliliter count of the sperms in semen, a whole sperm or a spermatid nucleus, so nucleus contains the paternal genetic material, it's directly injected into the cytoplasm of an oocyte that is removed from the woman's ovary. So there onwards the fertilization process will be continued. For insemination, ICSI needs only 1 sperm per oocyte. So here 1 sperm only is required per oocyte because a whole sperm or a nucleus is being injected to the cytoplasm. So here you can assume that the relative success percentage would be higher because already you are introducing this male nucleus into the cytoplasm of the oocyte. Unlike in conventional IVF, the sperm which will be inserted into a particular oocyte is already selected in ICSI. So there they choose a sperm before inserting it to the oocyte because only a single sperm is involved in this process. So to select that, they carry out a selection process to select a single undefective sperm. The fertilized egg can then be returned to the woman's uterus for implantation, similar to the IVF process. Now here I'll be teaching you some additional facts for your extra reference, and these will not be questioned in your A-level exam examinations. This is just for your knowledge. So first we'll be talking about the abortion process. So here this explains the dilation and the evacuation, abortion of a 23-week-old fetus. So here the first part, the body parts are grasped at random with a long tooth clamp. So this is the long tooth clamp. So they randomly grasp parts of the developing fetus's organs and they are pulled out part by part. The next step is the body parts are pulled from the fetal body out the vaginal canal. So here you can see a leg has been removed, an arm has been removed. So gradually these parts of the body are removed through this long tooth clamp through the vagina. So here they have also shown a 70 millimeter fetal forearm. So the third step is the remaining body parts are grasped out and pulled out. Here you can see the abdominal region, parts from the abdominal region are removed, and then the last the fourth part, the head is grasped and crushed in order to remove it from the vaginal canal. So this is the head. The head is relatively larger compared to other body organs, so it needs to be grasped and crushed so that it can be removed from the vaginal canal. And the final step is the placenta and the remaining contents are suctioned from the uterus. So here the uterus is completely clean after removing the placenta and the remaining structures of the developing fetus. So that's the abortion process, and this is not legally being carried out in Sri Lanka. So this prevents unwanted pregnancies. However, this cannot be done in Sri Lanka. Next we'll be talking about the proper use of a condom, the method of proper use of a condom. First, use a new condom for each sex act. Check package for date and damage. Tear and open carefully. So when obtaining this condom, you must make sure that no damage is caused to the condom. The condom is not torn or pierced from any region. Before any physical contact, place condom on tip of penis with roll rim facing away from the body. So in this manner, you must place the condom before any physical contact. Now, this condom has an ability of preventing transmission of sexually transmitted diseases. It doesn't have a 100% success rate, but it prevents this transmission of sexual transmitted diseases up to a certain extent. So before any physical contact, this condom should be placed. Then unroll condom all the way to the base of the penis. Now from here, it must be unrolled down to the bottom region. Now this must be done while the penis is erect. So after sexual intercourse and after ejaculation, hold the rim of the condom so it will not slip off, and withdraw penis from the vagina while still erect. Slide condom off, avoid spilling semen. So here, after ejaculation, you must hold it from the rim and withdraw the penis from the vagina without allowing any leakages into the vagina while all the semen is trapped inside this condom. Then throw away the used condom properly. After the use of this condom, you must insert it back into the pack which it came, this carton, and then dispose it properly. So that's the method of a proper use of the condom. And then the proper use of an oral contraceptive. Now Postino 2 is an example for an oral contraceptive. So first, take one 750 microgram tablet as soon as possible. That is like within 72 hours of unprotected sexual intercourse. So here you can see 2 tablets. Take one of this. Take a second 750 microgram tablet 12 hours after the first dose. Now here, when you are taking the first tablet, you must make sure that you will be able to take the second tablet after 12 hours, because sometimes you might be asleep after 12 hours, so you might be busy and you might forget to take this second tablet. So when taking this first tablet, you must make sure that you are free during the after 12 hours to take this second 750 microgram tablet. Now this here they have mentioned 7.75 milligrams, which is 750 micrograms. So each tablet is 750 micrograms, and this pack contains 2 tablets. If you vomit within 2 hours of taking a tablet, talk to your doctor for an additional tablet. So a substitute tablet must be taken to replace the one that was eliminated from the body during vomiting. So this can be identified as a defense mechanism by the body, because these oral contraceptives have synthetic or artificially produced estrogen and progesterone hormones. Postino 2 has an 85% chance of preventing pregnancy. Now that's a relatively high chance, but not 100%. There can be a 15% failure rate as well. If taken within 24 hours, there is a 95% chance of preventing the pregnancy. If taken after 40 to 72 hours, there is a 58% chance. So here you must make sure that you take this first tablet as soon as possible, because you can see if you delay the taking of this tablet, the success percentage reduces from 95 to 58%. Now that's a relatively drastic change. So make sure you take it within 24 hours. So immediately after intercourse is the best time, if possible. If you are able to take the second tablet 12 hours after the taking of the first tablet. It is not known whether it is effective if taken more than 72 hours after unprotected intercourse. So again, what it says is it's best if you take it as soon as possible after any unprotected act of sexual intercourse. So here, this is the posture 2 carton along with the 2 tablets. So that's the end of our video, and I hope you learned some important stuff that might come in handy during your exams, and also some additional stuff for your additional knowledge. And I would like to invite you to like and subscribe and share our YouTube channel and this video with among your friends, so that they also can learn something productive as you did after watching this video. And also don't forget to subscribe our YouTube channel and to click the bell icon, so that you'll be notified as soon as we upload a new video. So again, thank you very much for watching this video, and I wish you all the best and good luck to face your A-level examinations.